Labą dieną visiems čia susirinkusiam, aš Ingrida Domarkienė, pati esu genetikė, bet vystome paleogenomikos, archeogenomikos temą čia Vilniaus universitete. Ta galbūt matysime mūsų naujajame mokslo centre ir kūrėme komandą ir tam iš tikrųjų mums bekuriant gimė idėja pasikviesti čia profesorę iš Tartu universiteto, profesorę Kristiiną Tambets, kuri galėtų pasidalinti su mumis ir su mūsų bendruomenė savo patirtimi, koks buvo jų kelias ir ką galima nuveikti šioje srityje. Sritis yra tarpdisciplinė ir iš tikrųjų daug ką galima nuveikti su ta medžiaga, kurią turime ir pasitelkiant papildomus metodus, tokius kaip genetika, lingvistiniai metodai, archeologiniai metodai, istoriniai metodai. Iš tikrųjų visą tai mes galime sujungti ir gauti dar daugiau informacijos. Taigi, be ilgesnių įžangų, pristatysiu profesorę ir labai laukiame paskaitos. Tai profesorė yra pripažinta mokslininkė, jos įdirbis genetikos srityje yra neginčijamas, yra tarptautinės publikacijos projektai, kurie jungia tiek genetiką, tiek istoriją. Ji vadovauja archeogenetikos laboratorijoje, taip pat yra Tartu universiteto archeologijos, genetikos ir lingvistikos tarpdisciplininių studijų kolegijos valdybos narė. Taip pat yra aktyvi mokslinės bendruomenės narė, dalyvauja įvairių draugijų ir organizacijų veikloje, organizuoja partnerystės projektus, dalyvauja mokslo populiarinime ir yra išvertusi ne vieną knygą. Jos veikla buvo įvertinta ir vieno iš aukščiausių Estijos Respublikos apdovanojimų, tai 5 laipsnio Baltosios žvaigždės ordinų už nuopelnus valstybei. Tai iš tikrųjų turime galimybę pasikviesti, turime galimybę klausytis, tad tą ir darykime. Tai kviečiu profesorę.
Hello everyone, and I would like first to thank very much my colleagues from Vilnius University for this kind invitation to come and have an opportunity to share some of the scientific projects that we are doing currently in Estonia. And also, as I understood, the audience here is quite diverse. So I try to keep it simple and give you an overview about this field, which has undergone a rapid development during over last 40 years. And a couple of words about our institute. Our Institute of Genomics in University of Tartu is quite new one. And our institute next to the structural unit where I come from, which is Estonian Biocenter, which deals with ancient DNA research, hosts also Estonian Biobank. And I have to say that in Estonia, people trust very much science because almost 20% of our population have donated their DNA into the Estonian Biobank. And right now, my colleagues from Estonian Genome Center and Estonian Biobank are building a portal that allows to give feedback all those 200,000 people who have given their genes into Estonian Biobank. And it's quite challenging, as you understand, because we are a scientific institute and there are lots of challenges how this personal medicine information can get to the tables of family doctors, for example. And then we have also a core facility of genomics, which allows us to do the DNA sequencing in our institute, and also our ancient DNA data are produced locally in Tartu. And I am very proud of our clean room ancient DNA facility that now functions for 5 years already. Before that, because we had quite a big data sets of modern DNA from different parts of the world, it was kind of a context for appearing new ancient DNA data. But starting from 2018, we are capable of extracting DNA from the bioarchaeological material ourselves. And this is a big thing, I think, and I hope that our colleagues here in Vilnius University are also successful in establishing this kind of facility, and we are trying to be as much of help as possible. So the outline of my talk today, very briefly, is that I would like to give you an overview of the field. Then we have to start from somewhere, and basically with evolutionary genetics you can dig very deep, but let's first see what does it tell about the origin of our species, or Homo sapiens, anatomically modern humans like us. Then let's see what does it tell us about the origin of Europeans. And I have chosen here also some of the case studies, because when we established the lab, then of course we started the studies of prehistory of our own country, but I would say that this probably the take-home message is very similar also for most of the people, as well as people all over the Eastern Baltic, and also discuss some of the future perspectives of archaeogenomics. And as much as I understood Lithuanian, I think Ingrid also told you that couple of years in Tartu University we have had a collegium of archaeologists, geneticists and linguists. And I think that for successful collaboration within this transdisciplinary project, it is crucial, because we can start these projects all together from the scratch, from the beginning, and it has been already visible that it is the best way to move forward. And so some of the examples I give you here involve both genes, archaeology, and linguistics. So here is the tree, which is probably familiar for many of you, from Charles Darwin's sketchbook, where he tried to visualize basically the assumption that all living organisms today in the world, and also those who do not live here anymore, share the common ancestor. And this is the foundation of evolutionary genetics. So, and what does it mean? If we think about genes, then very often it's about some medical conditions, but there is much more information hidden into our genomes, and these are the ones that inform us about biological history of our species, but also the whole living world. And today, thanks to the development of technology, we have a great chance to study not only those organisms who live today and compare, for example, their genomes with each other, but also the organisms that once lived and does not live anymore. And this is the area where archaeogenetics get involved. Sometimes this field is called also paleogenetics, but in principle both of these words mean that we deal something that is very ancient. Is it just curiosity? With a couple of examples during this lecture, I tried to show you that there are very practical consequences of the knowledge of evolutionary genetics. And I think exactly 1 year ago, this field was, or one of the pioneers of the field, Svante Pääbo, was given Nobel Prize in physiology and medicine, which must probably talk itself. The fact itself says that this area is of big importance. And also what is important is that we live quite an extraordinary time, because right now there is only one species of human, but it hasn't been like this. In most of the time we have shared the same ecological niches or different ecological niches in the world with different hominin groups. And for a long time there was a question, how much did they contribute into our genomes? Do we still carry their heritage, or did they live separately from each other? And we come back to this fact after a couple of slides. And from one textbook that I love very much, which is Jobling, Tyler Smith and Kivisil 2012, basically there is a phylogenetic tree, again very simple one, which is based on the genomic sequences of different organisms. And what I would like to point you here is that if we study humans, then we study only the tipmost branches of this full diversity that exists in the form of tree of life in the world. And what it must tell us is that Homo sapiens is a very young species. It's extremely young. And it means that all the people who live in the world do not differ from each other more than just 0.2%, which means that whatever slides I show you and the differences between different groups, it's just such a small bit of changes in our genomes. And it also maybe shows you that we start to search for our roots, then there is like very different levels where we can go. We can study the shared root with, for example, brewer's yeast, but in today's talk we will stay in this tipmost branch of the tree of life. And as I told you with the example of collegium that we have in Tartu, this kind of studies are always very cross-disciplinary, interdisciplinary, transdisciplinary. And the thing is that the past that we have is the one and only, but the different evidences that we have for this past, they can inform us about very different aspects, this one and only past that we have. And all of these evidences that we get, both those that we get from archaeology or from linguistics or from history or from paleoclimatology or evolutionary genetics, they have their pluses and minuses. And let's say that the best results we will get if we know how to reasonably synthesize the results from different disciplines. And as you see from here on that picture, here is the timeline basically years ago, and here are different disciplines. You can see that with the help of different disciplines, we can reach into different depth of our past. And you see that here the evolutionary genetics and paleoclimatology go really deep into the past. Archaeology is a bit covers a bit smaller time range and starts probably from this time period when the first tools appear in the archaeological context, and also historical linguistics and history then cover the most recent time slices here. And of course, for any of those evidences that we can study, we know they are biased, because, for example, if we think about ancient DNA that we want to study, that actually these especially the older the bioarchaeological material gets, the less well it is preserved, and also the fossils are scarce and not divided equally at all in geographical space. And what we know already is that different environmental conditions are crucial. For example, it's much easier to get DNA out from permafrost than from tropical areas, and so on. And the same goes for any of those disciplines that we see here. For example, some tools preserve, the others not. Some languages have written text, the others doesn't. And all it means that this reasonable or meaningful synthesis of all this data is of crucial importance. And with genetic studies, we can study different layers of our past, and from the oldest to the newest, these are given here. And today we will concentrate on those layers that inform us about the origin of our species, discuss a bit the prehistorical migrations, and also some historical migrations. So my specialty is human and population genetics, and if we think about the archaeogenetic studies, then what we can reveal from those data is that we can see what kind of structure these ancient populations had. We can reconstruct migrations to see whether there is a continuity between different human groups who once lived and who live there today. How has it changed the population structure through time? Another aspect is that, for example, if we study some certain site of great importance, then we can see how these people who are buried, for example, in one cemetery, what kind of relations are between of those individuals. It means that we can study their kinship, and this is not only of studying their genetic background, but also it informs us about the past social life. And then one aspect that is studied with ancient DNA is paleoepidemiology. So we can study the spread of pathogens and also look for some disease-related or just it does not have to be disease-related genetic variants, but someone that is, for example, well visible, like phenotypic traits. We can trace their dynamics through time and see how it associates with environmental changes. And if we speak about humans, then also the cultural background and changes in lifestyle are very important to put next to these data. And we can also reconstruct, for example, the diet of past individuals. And this is when we do not study their bones directly, but use dental calculus that is preserved on their teeth, or then study the coprolites. And this allows us to look into the microbiomes of those ancient individuals. And these are only a couple of examples what this field allows us to do. And as I told, it's already almost 40 years when ancient DNA studies have been conducted, and the first one of those was on 1984 when in Alan Wilson's laboratory, and he is famous also for his theory about molecular clock. They were able to extract a very short fragment of DNA from one horse-like animal, which was called quagga. And at that time, when this sample was taken, this species was already got extinct, and the sample was taken from a museum specimen, which was approximately 100 years old. So how this has changed through time, you can see from here, because 10 years ago, this time was pushed back more than 700,000 years. And at that time, the oldest organism that was analyzed for ancient DNA was a horse from Canada and from permafrost. And it did not took much when this time limit was pushed even more. And on 2 years ago, there were even more than one individual who was more than 1 million years old. So the change has been very, very big. And what is behind it is that mostly it is a development of technology. And if I have had lectures for students, then almost always there is someone, and usually these are boys who ask, is it possible to sequence dinosaurs? Then this is hard to tell, because if we think about when dinosaurs lived, then there is a long way to go. And the other thing is that DNA as a biomolecule degrades in time, meaning that what we need here is that we need genomic sequence, but at one point it is just nucleotides scattered around, so there is probably some theoretical limit after what it is not possible to do these kind of analysis. So, and I think that dinosaurs are well behind this border. But this first, let's say, positive results of studying the extinct animals evoked a lot of interest. And here you see a couple of examples of the studies that are now called antediluvian ancient DNA studies, which all were proved after a while to be non-authentic. And as you see, the time what is given here is in a very, very distant past. And what was the reason for that? At that time, people were not aware of how serious is the problem of contamination, and why it was not contamination with modern DNA. And the other thing was that there was a lack of comparative data. If we compare the data sets that we have today for different organisms and what we had back at that time, then of course we even can't compare them. And it means that if you are able to get some DNA about what you do not know exactly what it is, then it's easy to think that it might be an insect in amber, because you don't have comparative data. But here is one of the example of dinosaur bone, which on the phylogenetic tree, as it is shown here, clusters together with humans, which means that this was at that time human contamination in that bone. And Patricia Bechnerova has described it in her blog post. The development of the field so that she illustrates it with a model that has been borrowed from USA technology company. It is called Gartner's hype cycle, and it is actually true for any new technology that is like first gives you groundbreaking results, that there must be some innovation trigger, and in our case it was then the extraction of DNA from museum specimens. Then there are a lot of let's say trials to sequence anything from anything, which is called here inflated expectations. Then it seems that no one can repeat these results, which is trough of disillusionment. But some of the groups still keep working on these materials, on these data, and they set some rules which all the people who deal with this technology must know. And this is called here the slope of enlightenment. And in the end, we end up in a plateau of productivity, which means that we produce new interesting results and data which are actually reliable. And here next you will see, you see also the explosion of human DNA full sequences, and it is this figure here is a couple of years old already. It comes from David Reich's web page from Harvard, and you see that the number of full sequences has grown very, very rapidly. And behind of it is basically the development of technology and the fact that starting from the beginning of 2000, there was a new technology which is called next generation sequencing, which is shown here with this red bar. And for those who are maybe not so familiar with all these technological stuff, it means that we can catch with this methodology ultra short DNA fragments. So before that, it was not possible. There were some time limits because the earlier technologies needed that this fragment had some certain size, but now they are like ultra short that can be caught. And it shows you that after this kind of technology started to be common, after that it took only a couple of years when the first genome, which was mammoth genome, was published in 2009, and not too distant from this is also the first human genome, ancient human genome from 2010, which was Paleo-Eskimo. And it's quite important why this particular individual was not, for example, of European origin, because the people who studied it were from Copenhagen, and if this was, let's say, at least in that stage when there was no ancient human genomes available, it was quite good proof that you can show that you are different than the Paleo-Eskimo. Then 2010, also Svante Pääbo's group published a genome data for our close sister group Neanderthals, and this is also for what partially he got his Nobel Prize. And after that also Denisovan individuals, and these two are then the close sister groups of modern humans. What is interesting maybe with this Denisovan individual who was found from one Siberian cave was that there was no actual full fossil available, but just a small fragment of finger bone. And sometimes it's said that this Denisovan is invented in laboratory, because archaeologists would tell that you need skeleton to study, but this was basically first only genomic sequence, and so on. And what then this methodology that allowed to catch these ultra-short DNA fragments meant, it meant that you can shift your samples that you study towards deeper time layers. And basically, it is true that you can extract DNA for almost any bioarchaeological material. Here are some of the examples, and if we study humans, then we always prefer the petrous bones from skull or then teeth, and teeth are especially good because when we extract DNA from teeth roots, then it allows us to study also the pathogens. And for example, if there are plague victims who died in an acute phase of the disease, then you can catch also the plague DNA out of those materials. And why here is this one mummy from Denmark, Graubalem, and this is because very often, if for an eye the biological material looks very good, then it's not always good for extracting biomolecules, and this is true for those bodies who were buried into the bogs, and there are many of them in Denmark because bog environment ruins DNA, and it's almost impossible to study there. So it means that if something looks good, it does not have to be good for studying ancient DNA, but usually if it looks bad, then it works bad. So this is like it is quite unfair correlation, I would say. And when this earlier picture, basically there are macrofossils to study available, but very often there is no macrofossils whatsoever. And there is also one field that studies sediment samples, and here is one example again from Svante Pääbo's group. Evelina Zavala extracted DNA from Denisovan cave sediments, and you see how well they cover the cave sediment with the samples, and probably it's not well visible, but they were capable to extract the DNAs which allowed to identify the hominin group and also put their relations with each other on the phylogenetic tree. And one new good source for ancient DNA is shown also here, and I show it proudly because my colleagues in Tartu University, and one of those is also Professor Aivar Kriiska, who gave lecture here in Vilnius yesterday, is about perch tar. And why this perch tar is important is that if we think about, for example, archaeological tools and then humans, and we find them, let's say, next to each other in the same time layer in some place, then you don't know, did this human who you found actually did the tool? But as this perch tar material was used as a gluing material, basically, to attach, let's say, if I am not an archaeologist, and I'm sorry if I put it roughly, that you have a hammerhead and a tail, and then you use the perch tar to glue them together. And what people did, they shoot them. So it means that they left their DNA there, and it's pretty sure that if you find this kind of perch tar and can extract the DNA, then you have a direct link between human and technology, meaning that the person who shot it most probably did this tool. And this gives us a lot of new information. And also the fact is that this perch tar was used a lot in Northern Eurasia during the Stone Age time, and we know that there is not so many of the samples from that time, so this is like a good materials to study different aspects in population history, about the microbiome of those people who shoot them, and also how the technology and humans are connected with each other. And this is just to show you, and actually when I once had a lecture where I had lots of quite simple slides, then my husband told me that you should put something complicated also here, that people see that you deal with serious things. And what I want to stress here, that of crucial importance in this work is also the bioinformatics, because like the DNA that we get from ancient materials is of poor quality compared to the modern DNA. Also, the bioinformatic analysis that has lots of tricks in it, and you have to first show that your DNA is authentic, and then to do these population genetic complicated analysis, you have to do some bioinformatic tricks with it. And as I told you already, you can study the organism who owns the bioarchaeological remains that you have. For example, if you have a teeth of a human, then you can extract human DNA from it and do your reconstructions. But you can extract also those organisms who colonized it during the macrofossil or its host during its lifetime, like microbiome or pathogens, or then also study the organism that colonized the remains after its death. And here is one of the examples from Maria Spirou's paper, which shows you the phylogenetic tree of plague that has put together from the sequences of different plague strains through time and different geographical locations. What we have learned now also from genetics is that we know that the cradle of Homo sapiens is in Africa, and what we also know is that, as I told it with the first slides of this lecture, our species is very young, and right now it is considered that it's around 300,000 years old, and we have actually lost Africa from the slide here, but I hope you know where it is located. And it is very hard to, or even impossible to pinpoint the exact region in Africa, because already in those early times there was probably quite complicated the population structure within Africa, and different groups mixed with each other, so it is hard to tell where exactly. But what we know is that around 50 to 70,000 years ago, some people left Africa and colonized the rest of the world step by step, and all these red arrows here indicate the so-called bottlenecks. And what it means that not all the people who represent all the diversity that was once in this place of origin did not move forward, and all the time there was like consecutive bottlenecks. And what we see today is that the genetic diversity in Africa is huge compared to the rest of the world. And most of our genetic heritage is then can be taken back to this out of Africa migration around 70,000 years ago. And recently there was a new paper in Science journal that, and I show it here because I wanted to show you that this graph here, what I showed you first, is really very simplified one. In this article, the scientist took almost 4,000 modern and some old, well-covered ancient genomes and tracked different genetic parts in our genome, and all these different parts, because in every generation our DNA are shuffled a bit, because we get half of it from mothers and half of it from fathers, and it continues back in time in between generations. So you can follow different genomic segments, and they all might have a bit different genealogies. And those genealogies, with the help of data scientists, are put here, so you see how messy it is. But what it shows here is that the deepest links here, which are shown with this darkest color, take us again back to Africa. And then there was a question that did our species or Homo sapiens had anything to do admixture-wise with these other sister groups of hominins of ours, because when they left Africa around 50 to 70 years ago, then they were not alone in Eurasia, and for sure they shared the different ecological niches with Neanderthals and Denisova people. And based on those genomic sequences that we have today, we see first that within the Neanderthal population there was also a structure. And what we also know today is that every one of us here in this audience carries from 1 to 2% of Neanderthal heritage. And this comes from the fact that after the out of Africa migration, we met those people. And it means that at that old times, Neanderthals and Homo sapiens had children together. And I know that this is a nice fact that those companies who give you a genetic test, they always sell it that you have 2% of Neanderthal, but you see that this is why we have it is in our biological or demographic history. And what is also known that the Denisova individuals have contributed to the people of East Asia and Oceania for around 4 to 6%. And what can be concluded here is that the admixture between those different groups was not unexpected at all. It was more as a rule than exception. And I think that the nice fact to know is that when we collect all those pieces of 2% from our genomes and put them together, then we get 40% of Neanderthal genome, which means that every one of us has a bit different pieces from this genome. And here is one of the examples which shows you that when you work in the field of ancient DNA, then you must have also some luck, because there is one of the representatives which is called Tenny, this 13-year-old girl who lived 90,000 years ago, and it is considered that her mother was Neanderthal and her father was Denissovan. So if we think about the scarceness of fossils, then it's a big miracle that someone found a specimen like this. And what does this contribution do in our genomes? I think we have lost some of the lines on the figures here, but maybe the take-home message is that many of those inherited pieces do nothing, and they are just the neutral variants. But there are also some that are connected today with, let's say, some risk alleles that some people carry. And one of the examples that are given here is that specific variant of this gene gives to the people who carry them a sunburn risk. And the others are the ones which are, so this is kind of a bad, if I may so, variant. But there are also others who are protective, and one that is shown here gives a protection against COVID, but there is another Neanderthal variant that makes people who carry it more vulnerable for COVID. So it's a very complicated system, but it definitely has an impact to modern humans, this heritage. And this is studied right now in the field that are called evolutionary medicine quite heavily. So when we come now back to modern humans and Europeans, then to understand it, how these historical events have left their traces into our genomes, we have to think about three most important events in our prehistory in Europe. And the first one is that at some point, a bit more than 40,000 years ago, there was a colonization of Europe by modern humans. And of course, this was not so simple as it is put here. It has several different details, but this is definitely a fact that we should know. And what happened after that was that last ice age came, which meant that, for example, the areas where we live today were covered with in its height, it was covered with ice that had 2 kilometers of thickness. So most probably people could not live at that time here, and they retreated back to the southern regions of Europe and recolonized then the more northern regions where the climate got better. And another very important event happened when there was a transition from hunting, gathering to farming, which happened in Near East around 11,000 years ago. And before, again, the genomic information was available, there was a constant debate about how did the farming spread. Was it more a technology that was learned by hunter-gatherers, or was it that the real people moved from Near East? And now we know that these early farmers really migrated and left their genetic traces into modern Europeans. And the third event is the Bronze Age migration that then took place around 5,000 years ago along the Ponto-Caspian steppes and left marks into our genomes. And I guess this is one of the most important papers that showed that almost every European today can be put together from the heritage of those three prehistorical events. And Wolfgang Haag in his paper has shown it here again with a very simple graph, which shows that the proportion of these different ancient components have different proportions depending on in which part of the Europe these individuals live. And as you see from here, Lithuania and Estonians are shown as someone who has the biggest contribution of this hunter-gatherer blue contribution in their genomes. This orange color shows the influence or input from early farmers, and you see that it is highest in Southern Europe and among the Sardinian population in this case. But also of big importance have had the steppe migration, which actually from our Lithuanian and Estonian genomes is the biggest one. And very often these complicated analyses are shown on a very simple, actually, genetic map. And as I'm going to show some of them in the following slides, I want to explain it a bit. So it's hard to see, but basically every dot is one individual, and with gray dots you can see modern Europeans and modern Near Easterns. No, at least I hope you see the clouds here. And with colored dots that are shown the ancient individuals. And as you see, they are very different in many ways from present-day individuals shown in gray colors. For example, European hunter-gatherers cluster here, early farmers cluster here, and then this steppe people cluster here. And what we see from here is that, again, all modern-day Europeans can be put together using these three big clouds here, and they are kind of in the middle of those. And the ones here shown a bit more darker color are modern Estonians, but I would say that modern Lithuanians do not differ too much from those. And the more close the dots are on this genetic map, the more similar are these two individuals also genetically. Why Eastern Baltics is interesting is that actually this very simple map shown here, and the blue color can be divided into more specific pieces. And what we know from the early prehistory of our area is that it was a meeting place of two different kinds of hunter-gatherers. One group was called Western hunter-gatherers, and they lived also in Iberian Peninsula but reached until the Eastern Baltic. And the other one was Eastern hunter-gatherers who lived basically here, and also they reached Baltics. And this is it is very important. And what I can say also is that from Estonia, we have data from Lithuania, thanks to this work that I show you, where also your colleague, Professor Jankauskas, was involved with his team. You can see here that this proportion in the genomes of different hunter-gatherers who lived our area changes. So who are down here, those show more Western hunter-gatherer input, and who are up here show more Eastern hunter-gatherer input. And also the input changes with the latitude. So what I can tell already now is that actually we don't know how did first individuals who lived in Estonian territory look like. And this is because we don't have bones from that time period. But when we extrapolate it from the more southern Baltic regions, then we could tell that they were similar to Western hunter-gatherers, like samples from Lithuania, but who knows? Let us see what the future brings if we are able to either find some bones or then extract the DNA from relevant places from the sediment. So this is one question that still probably will bring some new interesting details if we have more data. And then there is one paper of your colleague about modern population of Lithuania, but I took here some of the analyses that show with what kind of components are, let's say, most visible in modern Lithuanian population, and you see that the Western hunter-gatherer component among all the Europeans shows the hottest colors, which means that their presence here is the most well visible. And the same goes for Eastern hunter-gatherers, and also Estonians do quite good here, as you see. But here is the European early Neolithic, and you see that this hotspots in Sardinia and not so much up here. So these proportions of those different ancestry components are different in different parts is the take-home message, but basically all Europeans can be put together using this kind of puzzle pieces. And now I'm going to talk you a bit about Estonian prehistory, and you can take it as a little model how to study the local prehistory, because all these, let's say, works of or studies of European context or Eurasian context have now provided us, let's say, the insights how the general population has developed, but there are lots and lots of local details. And what we did in Tartu University when we had our own ancient DNA lab, we basically made a timeline of different things that happened through time culturally on our territory. And we can't go in Estonia, also in Lithuania, too much back in time, and it all starts, at least in Estonia, it starts with Mesolithic. In Lithuania, I have understood it comes also from late Paleolithic, but in general, we are speaking about 11 to 12,000 last years. And what has happened here during that time, that at some point in Neolithic, there was a transition from hunting-gathering lifestyle to farming. So one of the questions for us, as we did not have much samples from here, was that do we see any changes in the genomes of those two groups? One of those already did ceramics, comb ceramics. The others did corded ware ceramics, but comb ceramic people were hunter-gatherers, and corded ware people are considered to be already the farmers. But I will start here from another side of the story, because as I said, we have studied a lot also Estonian biobank data, and it is first very easy to study those people because they are still alive and they have a very good context. There are lots of information that are gathered through the questionnaires to know about these people, and why Estonians are different from other Europeans. When we think again about these three different ancestry components, is that we have something little in our genomes. It's about 5%, which comes from East, and which is not so much present in other Europeans. And also, if we compare ourselves with, for example, Latvians or Lithuanians, with whom we actually are genetically very, very similar, then what is clearly different is that we speak different languages. And here is the spread map of Uralic languages, into which also Estonian belongs. And what is believed is that the homeland, although this is heavily discussed, but this is not my expertise, so I have put the potential homeland here somewhere near the Uralic mountains, which gives also name to this linguistic family. So if we think that there is something in our genomes that comes from the East, then one of the questions is, did this come together with languages, these inputs into our genes? And of course, to put the languages and genes next to each other is not a new idea, because here is one of the books of Cavalli-Sforza from 1994, who did this kind of research very much, and this book is also sometimes called as a Bible of genetics. This part, what is interested about these questions that we discuss here. Although these are different people you see, who speak Uralic languages, if you look also phenotypically, they are quite different from each other. But still, one of the questions that we wanted to ask was that, do we have something which is shared by us with all those our linguistic relatives who live more distant from us? And what we found out when we studied the modern population of Estonia and compared it with modern populations of Europe that spoke Uralic languages and spoke Indo-European languages, then we found in our analysis, and this is one of the of this type of analysis, which uses basically genetic profiles. So Estonians are here pooled together into this group, but basically each stripe of this colorful carpet is a genetic profile of one individual, and it is visible probably for you that within one group these profiles are most similar to another individual from the same population. And Latvians and Lithuanians are here close to Estonians, and what we see is that they do not have this, or at least have it much less, this pink component that Estonians share with other populations who are living more east, which is shown here, yes, the pink color. And if we look at the geographical spread of this component, then this looks like this. What we also learned from the biobank data was that although Estonia is small and there is no big rivers or mountains, there is a clear regional structure in those data. And I have selected here also only two plots which compares the genetic closeness in different parts of Estonia with neighbors. And up here is the contacts between Estonians and Baltic populations, and you see, and this is of course expectable, that again this scale here is the warmer the color is, the bigger is the affinities between those groups. You see that it is southeast and south which shows the greatest affinities with the Latvian and Lithuanian populations. And if we compare Estonia with Finns, then you see that these affinities are visible in the northern parts. And actually these this is like, the structure is unexpectedly good, and it comes up if we have very well collected data, and these data have been selected so that we use, they are called old people, and these are 50+ people who live in countryside. And this kind of filter, if it was used for biobank data, it started to make very much sense because it excludes the recent migration between the towns, and then this structure came very well visible. So to conclude this part is that we have a clear population structure which is explainable by the contacts with neighbors, but also we have this eastern contribution that we share with other populations who speak the same languages with us. And now things come maybe also interesting for you, because if we look at the paternal lineages or Y chromosomes, which are inherited only from fathers to sons, they are very easily tracked in the genetic material in that sense that they do not recombine in different generations, and so they form in time and space a genealogical trees. And this is something in what Estonians, Latvians and Lithuanians are very similar of, because 1/3, basically this pie plot here is given for Estonians, but you can take it also as a proxy for Lithuanian population. And what I want to say is that 1/3 of Lithuanian men, like Estonian and Latvian men, carry the Y chromosomes that have definitely of Eastern Eurasian origin. And this particular type, and let's not go into the details of what its name is or something like that, you can see it's spread on the map with a blue color. And you see that the frequency of this type of Y chromosomes drops sharply, and it's mostly spread along the Arctic coast. And here is a quite sharp difference between Lithuania and Poland. So, and this is the question actually, how this particular pattern has arisen. There is no very good answer, but let's say in Estonian context, we often interpret it as some genetic impact which came together with the people who brought here Uralic languages, but how it has ended up so that we here within the Eastern Baltic speak so different languages but have so similar genes. This is now the question which actually could be studied together. Now the question for us, when we saw these kind of patterns in the genomes, in the full genomes and also in Y chromosomes, was that when did it come to the Baltic area. And for that, we needed to look into the ancient world again. And what we did was that, I will skip it and come back to it after a minute. And this is something that these questions actually came from humanities and came from our good colleague, Professor Walter Lang, who said that if you would like to see these changes appearing, please compare these two kinds of groups here. These are Bronze Age stone-kissed graves, which Walter Lang and his team has interpreted as something that has Scandinavian impact. And please compare it with Iron Age Tarrant graves, which have their roots, according to these interpretations, somewhere in the east. And if you look at those, they look also on the landscape very much different. And what was interesting for us was that if we extracted DNA from them, then we see that this red component that we started to see in Iron Age was not there in Bronze Age. So basically, of course, this is not the full representation of people who lived at that time in the Estonian territory, but we were able to see when this change occurred. And also, this comes for the full genome profiles, but what we saw here was that this eastern component was not visible in any of those men that we studied here, but was there in 15% of individuals that we studied here. So basically, the current situation that we see today in Estonian population and also Y chromosome-wise in Lithuanian population came, let's say, around 2,500 years ago. And we say at least 2,500 years ago during the transition phase, because we don't know, is it somewhere in some other Bronze Age community maybe already present? So this is the data that we have. And also, when we now take the full history of the Eastern Baltic, I would say, although this is given here maybe a bit Estonian-centristic, then you see that if these are here modern Estonians and Lithuanians and Latvians, then the first people who lived in Eastern Baltic looked like this. So you can see that they are very far away and very distant. So another cloud that I have made around is hunter-gatherers from comb ceramics context. And you see that there has occurred a shift within this hunter-gatherer cloud, but you definitely see that these people are also different from those ones. So now, with the onset of farming and together with coming of this steppe component, you see that the local population did again a shift, and the steppe and these people who were analyzed from Kordivere context looked like this. They are already quite close to modern populations, but very much different than the hunter-gatherers from comb ceramics context. So in Estonian territory and also from Lithuanian and Latvian territory, we have also data from Bronze Age, which shows that they make a shift towards this hunter-gatherer cloud again. But no, let's say these shifts are now around those our modern populations. And for Estonian, then we have also Iron Age, which takes us again closer to a modern population. So what is the take-home message here is that basically all of us are put together from the input of all these different changes that occurred, and we can tell that the cultural changes that we see in archaeological record were brought along by new waves of people, or actually it's more correct to put it vice versa. New people came and brought in the new cultures. So compared to any of other Europeans, we could tell that the Northern Baltic has this 5% in the full genomes that comes from the East. And what is also interesting is that when we turn to our colleagues from linguistics, then they tell to us that this was also the time when the Uralic languages came. So this is kind of a little nice case study which shows that in certain places, archaeology, genetics, and linguistics the evidence starts to tell you the same story. But I would say that the question about how these processes occurred in Southern Baltics is already another question, and for that I do not have you an answer today, but I hope that these new data that we probably will have also for Southern Baltics from Iron Age or the similar time period that we have managed to study in Estonia will bring some new cool knowledge about this. I have used all my time and I have some more slides, so I try to be here quick. One of the projects on what I am currently working, which is like very illustrative of a project that shows you how important it is to put together different data, is here. We call it Medesta, and it's about medieval Estonia, and it brings together all the knowledge that we can get from archaeology, anthropology, and also some environmental data to study the population history and disease history. And why this time period is important is that this is kind of a bridge between these our modern biobank data and also these prehistorical data. And these are the things on what we work currently. This is the map of the medieval samples with what we have covered the Estonian territory. And what I want to tell you here is that what we already have learned is that the structure that we see within Estonia has arisen in the early medieval times, because all these dots here are Estonian biobank data, and the colors correspond to the different geographical regions. And for example, if this is Saaremaa Island in the west, then our sample from medieval Saaremaa overlaps with this very well. But this is not the case with earlier times. Iron Age and Bronze Age Estonians are here, so they are far apart from it. But what we can say is that the structure that we see in biobank data was there already in the early medieval times. And maybe then one more example for you, and is about what is relevant for Kristiina and Ingrida when we discussed their plans of doing some ancient DNA work in Lithuania, is that we have studied the medieval time, also taken Tartu as one of the examples. And here are some of the churches from which the material comes, and one is interesting maybe because that this church, which was called St. Mary's Church, does not exist anymore, but at that place we have Tartu University main building, so all the skeletons come under this place here. But what we compared here is that we knew that the St. Mary people were regular parish individuals, were buried there. And then we have another context, which is Dome Church, which was definitely the burial place for elite. And we did not know what to expect, but what appeared here, it is not so well visible, but what we saw is that if we take the ancestry profiles of individuals from the parish cemetery of medieval times of Tartu, then they clustered together with rural sites and with modern Estonians, which means that basically these were people like us today in Estonia who were buried there. The ancestry profile is very much similar to this that we see today. But the skeletons that we were analyzed from the elite context from Dome Church, they clustered together with Western Europe. It means that in this medieval material, it's you can very well make difference of people with different origins. And these are the ones then who came from with Christianization from most probably from Germany. And these two groups are very well distinguishable. I guess if I may have 5 more minutes, then I don't have to skip this. So with that, I wanted to tell you that, for example, if you have similar sites here, then most probably it is very well visible, this different context where they come from. And what it also tells us that when we saw in the prehistory that those people who came had lots of to do with local population. So here it seems that those who brought in the Christianity did not have much to do with local people, because they look from their ancestry profile different here. And how my colleague, archaeology professor Heikki Valk has put it, he said that they came here not to live, but to die. So they came with their families and did not have much to do. Of course, there are exceptions, and these exceptions are very interesting ones, but let's not go into these details today. And as I said, with these materials, we can study also different pathogens, and this is one of the recent articles that is currently in bioarchive done by my colleague who works in our team, Marcel Keller, which is the plague genomes from Estonian medieval material, and not only in Estonia, but and these basically fall into this branch here. So the period soon after the Black Death was there in Europe, and what we found here is that this one of the oldest, or actually this is oldest from that plague outbreak individual we found from Otepää Plague Cemetery in southern Estonia. And what I want to conclude from here is that very many challenges that we see today in our society actually start to make sense if we think about our evolutionary or biological history. And there are different events in our deep evolutionary past, but also some of the very recent ones. And here are also, as you see, the Neanderthal and Denisova introgression that has brought along different kind of diseases that we see in present-day populations. And one thing what I want to point out here is that one of the big problems of nowadays society is, for example, obesity, and we start to understand immediately what is the reasons if we think about our evolutionary past. Namely, most of the time we have lived as hunter-gatherers, and the lifestyle changes that we have very recently have had such a big impact for us that, if I may put it so, the evolution has not worked as fastly to adapt us to the situation that we have today, that we can go to the refrigerator also at night time and have food, and there is lots of that. So this is one of the problems, that our genes are not yet there that could adapt to the current lifestyle that we live. And these kind of reasons we can find in many places. And what it also shows for us, and this is the last slide that I have for that, is that in Estonian biobank data, my colleagues tried to see how many of those traits that could be followed are associated with these certain different ancestry components. And they found out that from 27 that they looked, 11 made sense in that sense that you can connect them with those ancestry components and different variants. And for example, if we take only 2 of those, then we see that certain variants that come together with steppe ancestry are responsible of higher level of cholesterol, and the others which come together with hunter-gatherer ancestry come together with make our cholesterol levels lower, and some other examples. But what is important is that not only the proportion, but the quality of what kind of specific genes we have inherited is important. But of course, any of those traits cannot be used here to guess the dominant ancestry across one's genome, and also that maybe at some time in the past they did not have at all this kind of impact. And the future moves then, I think, in the archaeogenomic field there are kind of 2 directions. One is to synthesize all available data that you have. And for example, here is one elegant example from Rasimo's group from Copenhagen, where they try to see the reasons behind of those migrations. That they took, for example, big data from our knowledge of vegetation maps or climate maps, and you try to put them all together and see, can you tell why actually these migrations occur? And the other side is that you try to reconstruct concrete life trajectories. And I give you 2 examples here. One is what we did together with Dario Piombino, and these are Tartu University mummies. And the question here was that, are they real mummies? And very, very many different scientists contributed to this. And also we did some DNA analysis, which was quite poor one. But together all this body of data proved us that yes, really, these are real mummies from Egyptian period. And what I want to what I would like to conclude is that with all those, let's say, input from different disciplines, we start with this thing. but we end up with basically a living person. And skipping the conclusions here, I will have the last slide, which tells you that actually it is not about studying the old bones, but what we would like to reconstruct is the lives of the people who once lived and from whom we probably have a lot to learn. And with that, I would like to say big thanks to my colleagues from Vilnius University and from my team, and also these many other people who have contributed to all these projects from archaeology and linguistics. And also this is in Estonian, but it says Tartu University High Computing Cluster, which means that having a place where to do all your data science is also of extreme importance. Thank you very much, and I'm sorry if it took a bit longer. Yes. Yes, as I showed you, but this is again not my expertise. This, as I said, was the archaeology professor Walter Lang, who said that he sees really the shift, which shows that the earlier layer in archaeological culture was very much different than the one that appeared. So here, yes, the data come from this Tarrant grave context, which, as I understood, this particular context is made of stones, but similar things, they are called like dead houses, are quite common in southern Uralic region. So this is something that I can comment only as much as I did now. But yes, this was the reason why we wanted to compare this transition period between Bronze and Iron Age. So there are definitely some changes. And yeah, who knows what kind of context would be relevant, for example, for Southern Baltics. This is probably something that your colleagues are aware of or how to do it. So yes, I can comment that yes, this was pretty much not that we just blindly started to analyze them, but there was a hypothesis that was based on the context, archaeological context. Yes, over there.
So what did this Tarrant grave people wrote that the local population had to take the Nordic language? Why?
No, but this is, I guess, is again the question that if geneticists start to discuss about it, and then people who say, what would you know about these cultural changes there? So this is one of the, the most burning questions for Baltic area. And of course, when we, let's say, started and were young and saw these Y chromosomes, and then we thought that, of Eastern origin, for example, that came, then we thought, oh, maybe it was so that all people in Baltics spoke Uralic languages, and then Latvia and Lithuania started to speak Baltic languages, which we know is not true because Lithuanian language is considered one of the oldest in the area. So this is the question I can't answer well. How exactly these people who came were so, I don't know, cool ones that the local people take over their language, and moreover, language which was so different from the ones that are spoken now in the neighboring areas. So this is, yes, this we always think about it. What was it and how did it occur? So
is it the same with the Silent people from Norway, Finland? Are they also having the same shift?
Yeah, but this, they have, basically it is kind of a local study in that sense, that archaeology, linguistics and genetics in Eastern Baltic kind of show all the same things. But what I want to point out that, of course, people moved in different routes, and there are several of those. One was more southern, one was more northern. They moved along those several times. And for example, this Eastern contribution has been found already 3,500 years ago from the Scandinavia. But at that time, for sure, this kind of new wave did not bring in any new languages because they were not even split yet, these Sami languages from this common Uralic branch. So it is probably so that these routes of migrations were used several times, and sometimes they brought, let's say, some cultural changes and sometimes didn't. But it all makes very much sense in Northern Baltic context.
No more questions then. Thank you very much.